Okay, I started out as a student in microbiology with a minor in chemistry. And after I graduated, I did an internship. In that internship, I'm trained in a hospital, or I was trained in a hospital laboratory in all sections of hematology, chemistry, microbiology, blood bank, so on, so that I understood how to operate all the instrumentation, understood all of the testing, and then went on from there to work in reference laboratories, supervised um, and did other training for testing. And at some point, then we needed to increase the income. So I ended up going into sales and was fortunate enough I sold laboratory services to physicians' offices, but ended up selling products back to laboratories. I had a territory in the Southwest covering 5 states, and would go around to the hospitals and industrial laboratories and sell products that I had been trained to use back to the laboratories, and was able to do that from a position of knowledge and understanding of what the testing was about. Well, as far as understanding what a virus is as compared to a bacteria, I think everyone's familiar with bacterial infections, either a urinary tract infection, pneumonia, as opposed to having a cold, which is generally something that's upper respiratory. You have a cough and it goes away. That's a virus. And a pneumonia, typically, you can have a viral pneumonia, which is quite serious. But most pneumonias that people are familiar with are bacterial. Now, the difference between a bacteria and a virus, actually, bacteria have viruses. They're called bacteriophages. And in fact, a bacteriophage has been very useful for my brother-in-law, who got a Nobel Prize for developing a particular molecular species using bacteriophage infecting bacteria. So that gives you some idea how small viruses can be, because they're small enough to be a virus to a bacteria, which is quite small. I mean, the typical virus that we're looking at here with COVID-2, 19 for CoV2 is 100 to 150 nanometers. And that's, believe me, tiny. It's zero point zero 0.01, milliliters, which is very, very tiny. Anyway, so the virus particle gets in the air, it gets on the surfaces, and you pick it up, and you rub your face, or you pick your nose, or you do something around your face, and you inhale it. That's typically the way it comes in. It can also come in on food that, a viral particle really needs pretty high heat for an extended period to break it down. But a bacteria really is broken down much easier, and cooking, will of course take care of both of them, unless of course you don't cook your meat very well. But the bacteria typically is like if we eat it, it's because we haven't cooked our food properly. But a virus typically comes in through the respiratory system. And in with the SARS-CoV-2 virus, that's how we've thought of it. Uh, and it's got a particular mechanism for coming in. But there's been recent evidence that it's also ingested and can be entered the body through the gastrointestinal tract as well. Uh, Um, the virus particle is probably, let's say, 100 times smaller than a bacteria, a typical bacteria. I mean, there you can see, I can actually see a bacteria under a microscope. You can make a slide, you can stain it and look and find it under a microscope. You can't see a virus under a microscope.
Wow.
You have to use an electron microscope. And that picks up minute particles. And most of the pictures that you may have seen of viruses are an artistic drawing of what's been picked up by an electron microscope. And an electron microscope is you're shooting electrons at your specimen, and some of it goes through and some of it gets blocked by what's there. And so that you get an outline of what you're looking with. And then they further refined it and have been able to break it down chemically so that you can discover, what the various parts of the virus are made of. But
We see representations of the coronavirus, this round thing with these little protrusions sticking out. You really don't see that, right?
no,
You only see the outline of it.
yeah, I mean, I could I could show you. I don't know if you can make this out or not. That is an electron image that's been colored
Yeah.
of the virus particle, the SARS-CoV-2 particle. So that's, they don't say, but the enlargement of that is probably 500 times
Wow.
what they actually got out of it. As far as the particle size, that's probably hundreds of thousands of times
Oh, wow.
the size of the particle. Well, when a virus enters your body, it's actually quite fascinating, depending upon the particle, the virus itself. And God only knows how many there are. There's, hundreds of thousands of these things floating around. And just like, there's many bacteria, and most of which are not, you know, not a problem for us. But a virus particle comes in, and in this case, let me just focus in on the SARS-CoV-2, which causes COVID-19. And that particle comes in and enters into the cell through a particular doorway that's called ACE2. And so the ACE2 is a particular site on some of the cells in our body. And in this case, we'll look at the nasopharyngeal, breathing in through your nose. In those cells, they're on the edge of them, they have this particular doorway. It's called ACE2. And the virus particle comes in, attaches to that, and then melds into the cell through the cell wall. And once inside the cell, then takes over the mechanism of the cell to produce more virus particles. At some point, there's sufficient viral particles develop that it causes your cell, what used to be our cell, to explode, and more virus particles will come out into the system and spread. So that's typically how we do it. I mean, different virus particles use different doorways to get in. But basically, they need an attachment point, and then they use that to enter into our cell and use our cells to reproduce more virus particles. And it's all just genetic. It's RNA, genetic material that gets entered into our cells. And because of the programming there, allows for the takeover of our cells' processes, natural processes, and to instead produce more virus particles. Okay, RNA is ribonucleic acid. It's useful in different ways in our own body, but it's more, where we're interested in it is typical of these viral particles to have RNA in them, and that gets injected into our cells. Our cells function off of DNA. I mean, I'm trying to simplify this.
Yeah.
So our DNA determines what it is that we do. It's the message. It's the entire structure of our makeup is in our DNA. So that can be affected in the cells so that the RNA gets transcribed so that it looks like DNA to the cell, and the cell then functions as though that transcribed RNA is its DNA. And so it goes over that RNA and takes that message, and then, like a factory, starts making other viral particles.
Wow.
And they get put together and then spit back out into our bodies.
All right. Are viruses alive?
Well, it depends on what you mean by alive. I mean,
Well, I mean, or do they?
yeah, but
They
in terms of what we normally think of from a humanistic point of view or a human point of view, day to day, no,
no.
they are a conglomeration of molecules. But then again, so are we. And, I mean, that's all we are, unless, of course, you want to say there's some spiritual aspect.
Right.
Well, if we want to assert that, then what's to stop someone from asserting that there's a spiritual aspect to a virus that we just aren't aware of?
Okay.
So, is it alive in terms of our combating it? Yeah, it is. It's got a life because that life comes in and infringes on us, and it's like being attacked by a being. It is a being.
Uh-huh.
It doesn't have the kind of life that we ordinarily think of as life, but it certainly has a life that affects our life.
Okay, so it invades a cell somehow. It invades the cell, it penetrates the cell, and then it continues to reproduce inside the cell.
It uses our cells to reproduce more of it.
Inside the cell.
For instance, our cells or a bacteria cell
Yeah.
will reproduce itself. And so it'll keep multiplying itself. Anyway, a bacteria infection, the little bacteria keep reproducing themselves. Viral particles cannot reproduce themselves.
Oh.
They need our cells to reproduce other viral particles. So in that way, they're not alive. If that's what you mean by living, that it can reproduce itself, but it can reproduce itself by using our cells. Yeah, vaccines seem to not be well understood from beginning to end. but it's quite a process. It's quite a challenging scientific process. Uh, First of all, you've got to identify the organism. You have to know what it is you want to create a vaccine to. And then you have to look at it biochemically, see what the sites are on that particular organism, whether it's a virus, a bacteria, or what have you, and say, okay, this particular aspect of this organism seems to be available, that it would be what's called an antigenic site, which is something that the body, our body, and our immune system would recognize every time it saw it, and then have a reaction to it, forming antibodies, putting out T cells. We have our own specialized immune white cells, let's call them, that go out and they will attack invading unrecognized antigenic sites, or viruses, bacteria, what have you. So anyway, you have to identify a particular site. So that takes a little time. And then you've got to develop a method of making this antigenic molecule so that you can then take that to the next phase, which is to develop a molecule that you can inject into somebody and not cause harm. So how do you know that? Well, you do some testing on animals, and it seems that it doesn't kill them. Okay, that's great. And then you go on and you try that with human beings, and it doesn't kill them. Okay, that's great. Now let's do it on a few more human beings and see if it actually works. Does this attenuated molecule, in some instances, it's just like the first polio vaccine was an attenuated polio organism.
What do you mean by attenuated?
Attenuated, that means it's been inactivated, so it can't cause disease.
Oh,
It's been broken apart, or it's been damaged to sufficient degree that it can't activate its normal process of disease.
hmm.
So it's been attenuated, or that antigenic molecule has been produced somehow, and then you use that to try to develop an immune response. You put it into the human body and see if the immune system has an appropriate, useful response to that attenuated organism, or that antigenic molecule that you put in there, to see if that actually has a good response. Terrific. Let's say it does. Well, then you've got to move on from that, and you need to actually have a trial of it where people are given that, and then expose them to the disease after they've supposedly had the immune response and are protected. But you also need to have people who didn't get that, because you have to have a control study. You have to see whether it's just a placebo effect,
Right.
which means people think they're going to get help, or some other process going on. You have to eliminate that to make sure it's the vaccine that's working. So you test that on a large group of people that then end up being exposed to the disease. See if that works. If that works, then you probably have something, and you can go out and put it into use.
Hmm.
But it's a long process. It doesn't, the immune system takes time. Developing these molecules in the laboratory take time. It's not something that you just think up and then say, oh, okay, we'll do that. It'll work. These processes, you have to realize, are developed by very, very intelligent people who work for a very long time and stand on the uh, shoulders of other previous scientists who have done work. I mean, just think of how long it took to develop the Xerox machine. That's a simple thing. Okay, as far as using plasma, basically what you're doing is borrowing antibodies that were formed from a person who had contracted a particular disease. In this case, let's say they had COVID-19, and and their immune response, which is your body naturally sends out white cells to attack things, but it also creates antibodies, and it has two basic antibodies, IgM and IgG.
Explain to us in really simple terms what an antibody is.
Okay, an antibody is something that's specific for the particular organism. So you've seen the COVID-19 causative organism, and so you get an antibody that is created by your immune response, and it's a complicated process. Not going to go into it. Anyway. It creates something that will recognize that organism, and when it does, it goes up and attaches to it. And once it's attached to it, now it it keeps it at bay until you have a macrophage, which is a big white cell that comes in and eats all that up, destroys it, and that's helpful. So if you borrow somebody else's antibodies, in this case it would be IgG, which is a long-term protection antibody. It might be if it's soon enough IgM, but I doubt it. Anyway, if you get that long-term protection IgG antibody from somebody else and you put it into someone who's got the disease, those will go in and help to clear out the organism that's in there causing disease. And it does that by attaching to those organisms and holding them at bay until the macrophage, your own macrophage, can come up and eat them up. So it's, it's dicey, but it can work, and it can be helpful to keep somebody from having their own overreaction to a response, and that's a problem too. Is a overreaction in your own immune system to a huge infection, and that is something in the early days we were identifying that killed people because they would drown in their own liquids from having that immune response, over response. When you're getting the plasma, you're getting the other person's antibodies, and those antibodies the other person has developed to the organism that's causing your disease. And those antibodies will come in and help to clear out the organisms that are causing the disease you have right now. Okay, well, you asked me to see the difference between someone wearing a mask and someone not wearing a mask that is infected with CoV2, COVID-19 causing organism. And if it's colored, let's say red, and someone's not wearing a mask, then they're going to be kind of pink all over because they have been breathing, and every time you exhale, you are exhaling out these tiny particles. So it's going to be on their clothing. It's going to be like a little cloud around them. that as they breathe, it'll continually breathe out these droplets. I mean, they're basically going to be part of moisture droplets when they come out. Someone's got a mask on, and when they breathe, it's going to be caught in that mask. Depending on the quality of mask, there may be a little, pinkness showing up on the outside of the mask or none at all because it's captured by the mask entirely.
Right.
But they won't be infecting people around them. Now, the other person who's not wearing a mask is also probably touching their face and touching things. So they would, and they might cough into their hand and then touch something where they touched would now have a pink hue to it. And if they picked up something off the shelf and looked at it and then put it back, that would now have a little pink color to it. It's everywhere. I mean, essentially, we should be operating as though this virus is everywhere on everything.
Wow.
And whether everyone's wearing a mask or not, there's some there. If you go into Costco, for instance, and they are so picky and really good about it. There's already been people in there, however, who have undoubtedly, had the virus. And so it's settled in. And you still need to act as though there's some virus everywhere. And when you think about that, then you take it and you bring it home and you wipe it off, clean it off, or wash it off, preferably, and and then go on. But seriously, what's going on here that people aren't addressing is they aren't addressing the viral load, which just means how much virus are you taking in at any given time? Because your natural immunity is such that for very small loads, generally you may be able to take care of that without getting infected.
Mm.
You may be able to manage that. But if you go out repeatedly, if you go out without a mask, if you don't social distance, you're getting a huge viral load bringing in, and it will overwhelm your immune system. So, that's when you're pretty sure you're going to end up getting that disease.
So what is happening in these extreme situations, like in Michigan, where people are protesting and they're carrying their guns and they're may or may not be wearing masks and they're 1 foot apart and they're congregating. And if you could see that virus, what are you seeing?
Well, depending upon how many people are infected,
Right.
um, every time you've got a pink cloud surrounding the crowd. I mean, every time someone who's got it talks, they are expelling, um, particles of moisture. You don't see them,
Mm.
but in those particles of moisture, there's viral particles. Um, so, I mean, because this thing is really good at spreading. It reproduces quickly, and what is it, 2 to 3 days? I think you've got somebody expelling these things every time they talk to somebody. So they talk to this person, they talk to that person. How many people do they end up talking to?
Mm.
And if they shout, now they've expressed that up in the air. They've blown it up in the air, and it's now gets carried, to people all around them. And if
Right.
there's a wind, then that carries it even further.
Wow.
So, if there's 1 out of 10 people there that have that disease going on, then everybody in there essentially is going to be exposed to it. and
Right.
come down with it.
Okay, you mentioned that this is very good at spreading. How is this one different than, let's say, a regular virus, flu virus or whatever, that we get our shots for every year? Why is this one different?
It's really not, well, it is different in a unique way. What people are probably familiar with SARS or with
MERS?
MERS. I
Yeah.
mean, these are past coronavirus um, diseases that have come through, have been respiratory, and but they're not as virulent, and that is they don't cause as much harm as this one. This one is particularly virulent, and while we've had those, they've tended to die out. And they don't get picked up quite as well, and they aren't as virulent, so they don't um, really cause that much damage as they get spread. People will get laid up for a little while with a viral infection, and then it doesn't spread quite as well as this one does, and it doesn't get into the body um, as well as this one does. So um, this one spreads uh, in such a way that it is picked up and reproduced rapidly.
Hmm.
It's got a real rapid turnaround time, and it's also got a good staying power once it's expressed out into the air. But the real problem is that when it gets in you, it attacks those sites that I spoke about, the ACE2 site on many of the cell walls, like in your heart, in your lungs, and your nose. They even find it in your intestinal tract. They find it in your kidneys, and these are the places where you're finding problems, and it really messes with your body's normal function. Your body's a well-tuned organism, and it's got a lot of checks and balances. Like even with blood pressure, that's one of the problems. Is that it blocks the virus blocks the production of serotonin, which lowers your blood pressure. So
Hmm.
now you've got increased blood pressure, and this is why people with high blood pressure are some of the major victims of
At
this.
risk. Okay, so what makes this one? What what is the reason that this is more virulent than another? I mean, why is it bigger, or is it? I mean, what makes it bigger? I don't quite understand that part.
Yeah, well, I don't think anybody understands that part.
Oh,
Uh, that's that's just what it is,
okay.
and it's got certain abilities, and it's frankly beyond me at this point exactly what those are. Uh, but what makes it virulent is that um, it can get into so many different cells,
Okay.
and the kind of uh, response that it has, and using that ACE2 site makes it very difficult.
Hmm.
It's the specific site that it uses to get into our cells.
Wow.
As I mentioned, it causes a great deal of difficulty, and it causes clotting.
Wow.
And they're not exactly, at least I haven't seen anything where they've been clear about the clotting effect of this organism. But when it starts causing clotting, now you've got stroke as a problem. You've got kidney failure as a problem.
All right.
You've got heart failure as a problem. It's a, it's that's a biggie.
Okay, so Matthew, what could cause this virus and pandemic to stop if we don't find a vaccine? If there's another method, what might stop it?
Well, that's a good question. There's a number of different ways, and we're kind of in the middle of one of the ways, which is social distancing. Because if everybody actually completely separated themselves from one another, then the virus particle would die off as it was taken care of by one person, and once their immune system took care of it, that particular line of virus would die off, and that'd be the end of it.
Mm.
So that actually would work. Another way is to have massive testing so that everyone's tested that that can be tested, and we as we test people and see who it is that's had the disease or has the disease, and then trace back to their contacts and isolate those people. If we'd done that at the very beginning, we wouldn't have all the shutdown, which we should have done, but we were not prepared to do it.
Mm.
And we didn't, hurry up and get prepared once we knew about it. The other things that can happen, two other things. One is to develop a vaccine. And as I explained, it takes quite a while to develop a vaccine, particularly from scratch. Now, we've got some things going because some of the companies were already working on previous coronaviruses, other SARS virus like this one, although somewhat different, and they're going ahead trying with those, and other companies are trying to hurry up and develop one specifically for this virus, but that can take 2 years.
Wow.
And given some of the reports that we've had back from people in animal virus production, it's not encouraging about creating a virus for this a coronavirus.
Mm.
Now, another way, which seems to be much quicker in possibility at this point, is to develop a antiviral, what's called an antiviral. Now, there's two kinds of thinking on this antiviral process. One is that, as I talked about, that ACE2 site on your cells
Mm.
that the virus uses to get into your cells. If you develop what's called an antiviral, a molecule that goes in and ties up those A2 sites, then that keeps the um, virus from entering your cell and reproducing
Mm.
and establishing itself in your body. And that has some, possible side effect problems to it,
Wow.
as does anything. The other thing that they're proposing is that overwhelming the virus by putting in a whole bunch of molecules that look just like that ACE2
Mm.
site on your cell wall. So it's like a fake door, so that the virus will attach to that fake door, that molecule, and won't then infect you. It'll be taken out of activity with that. So those are the two things that I know of now, and I'm sure there's a number of other ones that are working on what they can do, but those are probably the most promising at this point.
If everyone social distance, let's say 10 feet apart, nobody got any closer than 10 feet apart from each other, right?
Right.
Eventually, the virus could not spread and it would die. But then some of the people have the virus, they need to go to the hospital, they need care. So in the perfect world, we're all separated out. Some people are just going to die anyway because they have the virus, right?
Oh,
So,
yes.
right. So then, but the ones who have it need care. Then you have a problem because they're in a hospital and the people caring for them, like nurses, doctors, and everybody else, are exposed to it and they're dying and they're getting sick. So I don't know if that's ever going to happen that way. But But I guess it would help if everybody social distance as much as possible then, right? And
Well, of course,
yeah,
yeah. But I mean, the problem that you present
yeah,
um, is in fact the way it is. However, it did not need to be that way.
right.
And we, I mean, we actually had the opportunity from previous SARS and MERS infections to get our self prepared for pandemic of this magnitude, and we didn't do it. And really, what you're talking about is a situation where we have healthcare workers who are fully gowned,
Mhm.
fully clothed, and protected
Right.
from having that virus come in and settle on them. Uh, and they can go in and treat these people and take care of them. And
Mhm.
if we're social distancing, it's not spreading, not that many people are going to get it.
Mhm.
And we can manage that with the existing healthcare system. But those healthcare workers and anyone, first responders, need to have the equipment to protect themselves from getting infected. And anyone who's been exposed to these people who are known to have the disease need to be isolated until the 14-day period or whatever they may want to put on them. uh, and once that's done, they can go back out because they would no longer be infectious themselves.
Mhm.
So it's really fairly simple and straightforward. It's logical and it's doable, and we didn't do it.
Yeah. Well, we really don't have the PPE and all the equipment that we need, like they're claiming we do have. We don't, right?
No, we don't.
That's
And
the problem.
we should have been stockpiling this so that when this first broke out,
Yeah.
and one of the biggest problems is the testing. I
Yeah.
mean, we didn't do the testing that we should have been doing.
Right.
I mean, they're the WHO organization, World Health Organization, and South Korea did the testing.
Hmm.
New Zealand
Right.
did the testing, and those instances, uh, New Zealand's a perfect example.
Right.
They had completely don't go out, stay home. Everybody was very compliant, and it died off. And they had the testing so that they could take people and take their contacts and isolate them and support them, through the disease process.
Right.
And they were able to come out of it, and we all should have been able to do that.
So, okay. Well, Matthew, uh, fantastic job.
Well, I hope it's hopeful. Yeah.